Hello, grade 8 students. In this video, we're going to continue with our digestion chapter, part 5, talking about the enzymes. So first, let's remember the neutral, the acidic, and the basic. We said that the neutral, the pH, it's approximately 7. It ranges between 6.5 and 7. Acidic, where the pH, it's less than 7. And here is the acidic. Whenever we move away from the 7, it's going to be the strong acid. For example, 1 and 2, they are strong acid, while 5 and 6, they are weak acids. Wherever basic, the pH, it's greater than 7. So as we move away from the 7 to 14 and 13, these are strong bases, like the household bleach and the hand soap, for example. However, the human blood, pH 7.4, it's considered as a weak base, while human urine is a weak acid, since it's less than 7, but it's near to the 7. So it's less than 7, so it's acidic, but it's near to the 7, so it's weak acid. However, the gastric juice, pH equal 2, so it's away from 7, so it's a strong acid. Keep in mind that digestive juices, except the bile, they contain enzymes. So all of them, they contain enzymes except the bile, which is created by the liver. So enzymes, they are biological scissors, like a scissor, but they are biological scissors. They cut, they break down, they digest, and they transform or hydrolyze the food. All of them, they have the same meaning, digestion of food. So these enzymes, they are recognized as a catalyst. What the meaning of this? This catalyst, it speeds up the reaction of digestion. It makes the digestion process faster, but it will remain unchanged. The quantity of this enzyme will remain the same at the end of the reaction of the digestion. So moving to this table is very important. Already we started with this table, starting with the first table. We started about digestive glands, organs, digestive juices, enzymes, substrate, product, medium, and the temperature. As you notice here, all the temperature, they are 37 degrees Celsius. What does this mean? This is the normal body temperature, and the enzymes, they can act, they are optimum temperature, which is meaning the best temperature. In the mouth, we have a gland which is called salivary gland. The salivary gland, it secretes a juice. It's called saliva. This juice contains an enzyme called salivary amylase. So salivary amylase, it works on a specific substrate. We call it the starch. So salivary amylase works on starch to give maltose. The medium is neutral, where the pH is approximately 7. And the temperature is 37 degrees Celsius. So this is the saliva, as we said before. It's found inside the mouth, and it contains the enzyme salivary amylase that digests starch to give maltose. That's why if we put a piece of bread inside our mouth, this piece will become sweet in taste due to the change into maltose. Second, inside the stomach, we have a special gland called gastric gland. It secretes a special juice called gastric juice. Contains the enzyme pepsin that works on protein to give peptide, and the medium is acidic, so the pH it's less than 7, and sure temperature is 37 degrees Celsius. So here the stomach, we have a special formula. We have the triple P, P, pepsin start with the letter P, protein start with the letter P, peptide start with the letter P. So pepsin works on protein to give peptide, so it's P. Protein is the complex form of protein, while peptide is the intermediate. So here we have the pH level of the stomach. The stomach resting pH is about 4 to 5. After high protein, it drops to 1, 2. Now, let's move to pancreas. Contains a juice. It's called peptidase. So any word that ends with the A's, A-S-E, from now on is going to be an enzyme, like the case amylase. So it's also an enzyme. Peptidase, as its name indicates, peptidase, it digests peptide. So into amino acid, which is the simplest form of protein, where the medium is basic. So meaning that pH is greater than 7. And the temperature is 37 degrees Celsius. As you notice here, only the mouth and the stomach, they are not basic, while the rest, all of them, they are basic. Only the mouth is neutral, and only the stomach is acidic, while the rest, all of them, they work in a basic medium. So this is the pancreas and secretes the pancreatic. Also the pancreas secrete pancreatic juice. And the pancreatic juice, we have other enzymes called trypsin. So it works on protein to give amino acids. So we move from the complex form to the simplest form of protein. Amino acid, medium is basic, temperature 37 degrees Celsius. Also the pancreas, it has a special juice, pancreatic juice, contains an enzyme amylase. Wait a minute, amylase? We heard about this before. Here's another amylase, salivary amylase. The salivary amylase, it works inside the mouth, so the medium is neutral. While this amylase, it works at the level of the small intestine, okay? Where it works also on starch to give maltose, but the medium here is basic, while here the medium is neutral, and always temperature is 37 degrees Celsius. Also, pancreas at the level of the small intestine secretes pancreatic juice. This juice contains another enzyme called lipase, so ends with the word ase. Lipase, it works on lipids to give fatty acids and glycerol. So here, lipase, it digests lipids to give fatty acids and glycerol. The medium is basic and temperature 37 degrees Celsius. As you see here, lipase works on lipids. For example, peptidase, it works on peptides. And last but not least, we have liver, secretes juice that contains bile. Wait a minute, here we don't have an enzyme. Oh, we remember the note. The bile is the only juice that contains no enzyme. So what is the role of the bile? It works on lipids, but it doesn't digest lipids and fatty acid and glycerol. It leads to emulsification of lipids. So it digests or it cuts or breaks down the lipids into small pieces. These are called emulsified lipids. Medium is basic and temperature is 37. So for lipase to digest the lipids into fatty acid, we need the action of bile. So now let's move also to the level of the small intestine, but we are going to move to the gland which is called the intestinal gland. Intestinal gland, it has special juice. It's called intestinal juice. This intestinal juice have an enzyme called sucrase. So it ends with the word ase. It ends with ase, so it's an enzyme. Sucrase, it works on sucrose to give what? What are the products? Glucose and fructose. The medium is basic, pH greater than 7, and temperature it's 37 degrees Celsius. So it digests sucrose into, this is sucrose, it's a glucose plus fructose. So the green one is glucose and the violet one is fructose. While intestinal gland at the level of the small intestine, they secrete a juice also called intestinal juice that contains the enzyme maltase. So it ends word ase. Maltase, it works on maltose. The product or the medium is basic, but the product is going to be glucose and glucose. All of you, you know that maltose is going to be digested at the level of the small intestine. So it will give final product to glucose molecule. Medium basic, meaning that the temperature is 37 and the basic pH greater than 7. As you see, all the temperature they are 37. It's the optimum temperature. It's the best temperature for the working of the enzymes. So here, maltose by the enzyme maltase. This is the reaction. It gives two glucose and glucose. While also at the level of the small intestine, we have intestinal gland secreted juice, which is called intestinal juice, that contains the enzyme lactase. Lactase, it works on what? On lactose. So they are very simple. Sucrase works on sucrose. Maltase works on maltose. Lactase, it works on lactose. And these, already we studied them, that lactose, when it's digested, it can give the glucose and galactose. Medium basic, pH greater than 7, temperature 37 degrees Celsius. So here, lactose by the enzyme lactase, it can give glucose and galactose. Okay, so this is what is known by the action of the enzyme lactase. Finally, we have intestinal glands, intestinal juice, and they have the enzyme lipase. It works on lipids also to give fatty acid and glycerol, where the medium is basic and temperature is 37 degrees Celsius. This is another lipase. So this is the process of the emulsification of fats. Emulsification of fats is done by the action of the bile. Bile can digest the fats, the large lipid molecules, into smaller ones. These smaller ones, they can be digested by the action of lipase. So lipase break down small particles, small fats, into glycerol and fatty acid, while lipase can digest the large fat. And here's the importance of bile. Bile, it can cause the emulsification, it causes the segregation or separation of large into smaller ones so that they can be digested easily by lipase into fatty acids and glycerol. Thank you all. Bye bye.